I'm Kavya. Here we are going to see about apomixis. Introduction. Apomixis refers to the development of seed without sexual fusion. In apomixis, embryo develop without fertilization. Apomixis derived from Greek word that is apo, away from, mixes, act of mixing or mingling. Apomixis is identified over 300 species, most frequently found in families like Compositae, Rosaceae, Gramiae, Asteraceae. Apomixis is the most powerful breeding tool to fix heterosis, but it is nuisance when breeder decides to obtain sexual progeny because recombination of crossing is impossible. Agamospermy. In this category of apomixis, the plants have retained seed as the agent of propagation, but the embryo is formed by some process in which normal meiosis and syngamy have been eliminated. This phenomenon is known as agamospermy. There are three different types of agamospermy. They are adentoembryony, diplospory, and then apospory. Attentive embryony. In this, embryo develop directly from the diploid sporophytic cells either of nucleus or integument. The sexual embryo sac develop in normal way and the zygotic embryo either degenerate or competes with the apoptotic embryos. Diplospory. A diploid embryo sac is formed from a megaspore mother cell without a regular meiotic division. Example, Arriva tomentosa. In this type, an archisporium differentiates, but the megaspore mother cell develops into an unreduced embryo sac. The embryo formed by the unfertilized egg, that is parthenogenesis, or some other cell of the embryo sac. They are apogamity. Apospory. This phenomenon was reported in angiosperm for the first time by Rosenberg in 1907. Here, a somatic cell in nucleus directly from an unreduced embryo sac and the diploid egg parthenogenetically develop into embryo. Here, the megaspore gradually degenerate and caposporic embryo sac may develop from a functional megaspore mother cell. Dr. P. Maheshwari in 1950 has subdivided apomixis into three types. They are non-current apomixis, recurrent apomixis, and then addentive apomixis. Non-current apomixis. The megaspore mother cell undergoes the usual meiotic division and a haploid embryo sac is formed. Hence, the embryo arises either from the egg or from some other cell of the gametophyte. They are haploid apogamy. This type of apomixis has been seen in several species such as Solanum nigrum, Nicotiana tabacum, etc. This is the development of embryo sac. Recurrent apomixis. In this type, embryo sac generally arises either from an archisporial cell or some other part of the nucleus, and all the nuclei of the embryo sac are diploid and there is no meiotic division. The embryo arises either from the egg or from some other cell of the gametophyte. Generative apospory has been observed in Euphorbium glandulosum and then somatic apospory has been reported in Hierarchia excellens. Attentive embryony. This type of apomixis is also known as sporophytic budding. Development of embryo sac may be haploid or diploid, but the embryo do not arise from the nucleus or the integument. There is no alternation of generation, but the diploid tissues of the present sporophytic directly give rise to the new embryo. Attentive embryony has been frequently reported in citrus, Euphorbia dulcis, Mangifera indica, and some other examples. And in significance, the most important apomictic crop plants are citrus, mango, and blackberries. This reproductive system, it offers the possibility of the indefinite propagation of specially favorable biotypes, which may be highly heterozygous or sexually sterile. Thank you.